Good
Hello.
morning, good afternoon, evening, depending on which time zone you're in.
Nice to see you again, Ron. I appreciate you being here.
Same to you, Ben.
So if you wouldn't mind, as we've been going through this, provide us with a little bit of background bio so that we know exactly the expertise from which you'll be speaking to us today.
Okay, and expertise we will put in quotes for the time being. Thank you. So I'm a relatively newcomer to this space based on our last two speakers. I've got over 2 decades of experience, which is clearly just a fraction compared to to our last two speakers who gave some fantastic presentations. Um, I started off actually, so I'm a CMC experienced person. I've been in everything there is to do with CMC for large molecules, a small bit for small molecules as well, dipping my toe into that for a little while until I realized that I don't like methylene chloride headaches and went back over to the large molecule space where I do like aqueous water solutions. So, there we go there. I've worked in cell line development all the way through auto injector development and implementation. I've licensed a number of molecules. I've been on development organizations and commercial organizations, filed many supplements for next generation molecules as well, mostly in the monoclonal antibody space or FC fusion space. I've also worked a little bit with E. coli and some anaerobic cell lines, which are pretty nasty. And again, I come back to I like our antibody, monoclonal antibody, and chill cell space because everything is really nice and well designed. I've worked at big companies and small, Pfizer, Bristol Myers Squibb, Allergan, for example. And my current small company is at Immunovant, which is a virtual company located out of Manhattan, but really located all over the globe because we do our laboratory and manufacturing work at CMOs and CDMOs and CROs. So that's a little bit about myself.
Great. So you're uniquely qualified to speak on this exact topic then. And thanks for joining me today.
All right, thank you.
So you endeavor to talk about increased bioburden excursions in an aging facility, which is the title of your case study. So, I guess to sort of color the background, aging facilities obviously have their challenges. They can have different challenges. There's always a certain character that comes with different aging facilities. So please give us some of that context for the facility that you'll be speaking about.
Yeah, of course. So just to complete the context picture, I am talking about a fixed stainless steel type of facility, not an old disposable facility, maybe like what Paris was talking about back in the days of Accelerex. But this is a very old, let me change that a little bit. This is an aging facility that has stainless steel mostly. And one of the biggest problems there, you kind of got what you got. So if you want to do process A or process B or process C, you still have the same facility, the same piping diagrams, the same flow paths, the same tanks. So you do, you do only have what's at your fingertips or your disposal. So sometimes you need to make some slight modifications. Sometimes, over time, those modifications turn into something that may not work as well as you thought it would. And, in today's world, especially the quality and the regulatory aspects, there's a ridiculous amount of scrutiny on bioburden and endotoxin hits, even in drug substance manufacturing, not just drug product or sterile manufacturing. And if you're looking at, environmental monitoring hits in floors and walls, not just on equipment or in process streams. So what you have to make sure you do is your whole facility is under the same umbrella of, and this is even in closed processing. I'm not talking about open processing, but just closed processing systems. So you really have to make sure that you have, complete control on the environmental systems surrounding and in your process. So that's kind of where we're going to begin the talk.
Great. Yeah, I appreciate that. So within that context then of the facility, what was your challenge exactly?
Well, there's some, my timer started here, sorry. So there was a number of challenges, but the biggest one was eliminating, I like to use the word eliminating, even though the reality is we're minimizing, but eliminating the microbial hits, be it bioburden or endotoxin. And the reason that's important, as I mentioned, is all the regulatory scrutiny. But the other things that come to mind, it is a huge time sink, as you have to do investigations to figure out why you got an environmental hit. It's a cost sink if you have to throw away batches, even if it's batches of media or buffer, or you have to repack chromatography columns, or worst case scenario, you have to throw away batches of drug product or drug substance. So it's really important to make sure that you have a facility that doesn't have a lot of these environmental hits. Unfortunately, in aging facilities, modifications are constantly happening. As we bring in new equipment, as we change flow patterns or flow paths, we replace floors. Often you just add an extra layer on top of your existing floor. You don't actually go down to the foundation. So you have issues there where water can get trapped and molds can form and things. You also change room layouts a lot. So I mentioned, different processes come in. Maybe you need to change the configurations. Occasionally, these will lead to unforeseen negative consequences. Today, we're talking about negative or positive bioburden and endotoxin hits. You want to pop the next slide, if you don't mind. So I just wanted to throw some examples. In dogs, are diaphragm, O-rings, and gaskets. Just shortened it to dogs because it's kind of catchy. So here you see a bunch of some diaphragms and some O-rings and stuff, and you can just see and if you click on that thing that they talked about earlier, that little diamond thing.
Yeah, it'll expand.
You can actually see that in a little bit more, but you can just see the tears and the rips in these O-rings, and these are actually coming out of process vessels. So you can imagine trying to hold back liquid, sometimes 4,000 or 5,000 liters, and this is your only barrier. So there have been some hits on this. Take the next slide, please. The other thing that ends up happening is you start looking at flow paths that lead to unwanted or unforeseen consequences, such as dead legs and in places where things can pool. And here you see a little tiny double arrow that shows that even if you're flowing your cleaning solutions, there is a little place where liquid can pool. And you guys can't see me pointing to that, I apologize, but trust me, I'm pointing to that little double arrow there. Those are areas that all of a sudden, no matter which way you flow, no matter how fast you flow, you're going to get an area where the liquid may or may not come in contact with steam or with your caustic solution, for example. Go to the next one. So the other thing with aging facilities is often, you just kind of tack on to what you get. And, maybe at one point this was only going in one direction, but now you're looking at two directions. And depending on which way you're flowing, you're always going to have a dead leg again. So again, not much you can do. I think that was my last slide, Ben.
Yeah. So you got this one here.
Oh, here's another one. So this is an example of a vertical dead leg instead of a horizontal one. Here the steam can come in. It can go to one or two valves. Either way, you're only using one at a time. You're going to have a dead leg. So these were unintended consequences from design, not failures, but just maybe not necessarily having the high quality engineers that you need. Okay. So that's kind of setting the stage for where we are and why we get environmental hits in some of these facilities.
Yeah, and I think, you mentioned earlier eliminating versus minimizing. I think eliminating is the right word because it's like OSHA recordables for safety.
Yeah.
You strive for zero, even if zero is statistically impossible, because as soon as you say minimizing, it sounds like you're capitulating to eventualities in the facility. Well, we'll minimize as best we can, which sort of means we tacitly agree that if it costs too much, we won't fix it.
Yeah, totally agree. Yeah. And that's the way the regulatory bodies respond to those questions too.
Yeah, exactly. So the options then, and I really appreciate the forensic photography there of what could go wrong and where you've seen these issues. So what options did you face for your facility?
Yeah, so the other thing you have to take in mind, whenever you do any major modifications to your cleaning or to your piping or your flow systems of any kind, you in a validated system, you run into the problem of maybe having to revalidate. So there's a number of things that you do. The first thing is you just increase your cleaning. If I used to clean for 10 minutes, maybe I clean for 20 minutes, maybe I clean for an hour. You also could look at changing your current practices, adding a different caustic, adding a higher normality caustic, increasing your steam temperature, changing the flow rates, things like that. And then another option is to actually change your facility, as you mentioned. We see some of these dead legs, cut them out and replumb them. And then another option you can do in regards to facility modifications is maybe you don't even need those stainless steel lines anymore. Maybe you go to plastic or disposable type tubes where you really eliminate the potential. Again, theoretically, you eliminate the potential for microbial growth because you're tossing those after every batch or every couple batches or whatever your cadence is. So again, minimize the opportunity for liquid to build up and bacteria or molds to grow. In our case, we looked at this, backwards and front ways, and we did what every company that is currently doing a campaign does, as we start with the simplest. I got a hit, let me re-clean. Let me do whatever I can to get it ready for the next batch. And some of those times it worked, some of it didn't. Another quick option, maybe you have a little bit longer, but not an eternity, is just to change out your dogs, right? Change all your diaphragms and your valves, change all the O-rings and gaskets in your tanks and your tubes and on your valves, and really just look. Maybe some of them look like some of the pictures I showed you, and you're like, oh, thank God I got rid of them. The other is to change your exposure time. If you're doing steam for a half hour, do it for 45 minutes. Flush volumes. If you're flushing a series of pipes and tanks, instead of 1x, do 2x or 3x. And of course, maybe use higher pressure steam. Or if you're using hot Wi-Fi, maybe use, maybe heat up the tank or something on top of that too. Again, do what you can do as fast as you can to get ready for the next batch. That's often what everyone has to do. And during that, the other problem that comes out is you may actually go outside of your validated range. And if you do that, you got to redo your cleaning validation. Okay? Right. So, that's like step one. If you start seeing a couple of repetitive ones, then it could get kind of nasty, and then you got to look at doing something a little different. And that's where facility modification really comes in. It's really easy for us to look at those pictures and say, well, just snip it, maybe slope it so that there's no possibility of a dead leg on one side. There's what seem like simple fix. However, often in some of these old facilities, this is one line and in a matter of 100. This one got highlighted because it had a hit, but maybe there's 99 others that don't. So you do have to really kind of keep working your way through and figure out what makes the most sense for your entire facility. Sorry, excuse me. And then, the other question that really comes to mind is can you actually reach them? Some of these facilities have just grown and grown and grown and grown, and you need to change one that's in the back. You've got almost no shot of doing that because now you've got a big Wi-Fi still in the middle of it, or a couple process tanks, or they built a mezzanine where to do it you got to crawl on your back, and contort yourself like a gymnast in order just to get in. And you've got to shut down your facility because you got to bring in cutters and welders and all the other stuff. Then you've got a time where you got to re-clean. And so sometimes facility modifications don't work, but those are the ways to eliminate, not to, hopefully minimize. So that's some of the stuff that we did. The last part of that, as you can imagine, is if I'm cutting it out, do I really want to replace it back with stainless? Do I want to come into the same problem in 5-10 years, or is there a way I can avoid that? Maybe if I just kind of replumb things a little bit different, I can go ahead and use disposable tubes, disposable bags, disposable factors.
Yeah, so along those lines, maybe there's another option too. In pre-soliciting questions, we've got a question from an audience member prior to the event, and that's where does staff training and qualification for aseptic processing fall in the mix?
Yeah, that's a good question. So the assumption that I made for this talk, as well as for the plan I was in, that training was done as part of your GMP training. So staff understood, the following the batch record was mandatory, for the following reason. And one of those, if it was a cleaning batch record, one of those was for aseptic processing to ensure sterility, to ensure, microbial death. Just like as much as turning a valve for a chromatography column was needed for GMP reasons. You had to do it at the exact same time every time, according to the batch record. So aseptic training was mainly assumed. If you had an open connection, you were in a fume hood, a bio hood, or you were in the necessary room qualification for that, and you were trained on the aseptic nature of that prior to doing the activity. So essentially, the answer to the question is, if you're not aseptically trained, you really shouldn't be doing these operations. And in a GMP manufacturing facility, you could get cited for that. In fact, you would get cited from your local quality unit.
Mm. So your selection then, out of all those potential options of increasing cleaning, modify or changing the cleaning practices, implementing facility modifications, aseptic training, is really to ask the question, okay, to do it right, we modify the facility, but do we move from stainless steel to single-use connections?
Yeah, I mean so that's the ultimate question. You listed out as you were running through them the risk assessment that we would do, or the 5 whys, depending on how you would look at an investigation, to really figure out what's the best. There are rare occasions where it's just, wow, we didn't clean well enough. We really know that for whatever reason. Then we go ahead and modify the cleaning procedure essentially, based on the data we had. But the reality is that's usually a small term fix or a short term fix. And then you're back to the other part of the question, which is, what do I do once I decide I got to modify the facility? And for us at the facility we're talking about, the answer was really clear. There were some parts of the facility that it was just a no-go. But other parts of the facility, wherever possible, bring in a disposable tank to replace an aging stainless steel one. Bring in tubes that connected, bags of buffers directly to the skids, the chromatography, the filtration skids, or the media to the bioreactors, things like that. Those made a lot of sense. The other thing that became really obvious, we have a lot of hits in the inoculum room where we were doing transfers that are really so small, and I mean, relatively speaking, that it fits incredibly nicely with the disposable space. 50-liter bio bags and all these connectors, as opposed to a 50-liter stainless steel portable tank and legs. And those are the things that were just really easy to say, this doesn't make sense. Let's go disposable wherever possible. There is a cost to disposables, and I don't necessarily just mean the monetary. You do have to make sure you have the warehouse spacing. And as was mentioned earlier today, supply chain is getting kind of crunched on different things, one of which is the availability of plastics. So bags, connectors, filters, the things that increase use in a disposable facility or disposable components in general, you do have to make sure in today's world you have the warehouse space to stock up on those.
Yeah.
But nonetheless, it is one of those questions where you just go down the row and you do what makes the most sense for you, especially if you have a little bit of money in your budget and you have a little bit of forward thinking from management to allow yourself to make these hard choices to say, I'm going to cut out tank A, B, and C and put in 3 bio bags, or facilities to handle 3 bio bags, 3 single-use mixer, or a single-use mixer and a couple of bio bag holding tanks.
Yeah, in fact, just like at the outset, it's organization and strategic planning, it's leadership, trying to come up with what are the right things to do to not maintain the status quo, but to get better. And it sounds like this has definitely worked for you. There was one more question that came in too, that I guess it's more of a claim, but it is, do you think moving from stainless will improve the reduction in bioburden, when actually in reality it's the other way around? People, process, material movement are the major source of increasing bioburden.
Yeah, it's each facility is different, and some of the pictures I showed you in a few other cases, the facility was the big root cause. We could have put stainless steel back up, but again, in today's world, sometimes you do, sometimes you don't. I'm a firm believer that if you do things right, it doesn't matter what your facility or what your systems are. However, I'm also a realist, and no matter what happens, nature will find a way. So if I can minimize that, I'm going to minimize that. And if I switch from stainless to disposables, I don't have the continued use, the continued storage, and should be dry, but yeah, there's a little bit of liquid in there, and now I got a biofilm form. So that's the reason that we selected for the most cases, change the facility, don't put stainless back, put disposables. But it is a case by case. Like I said, sometimes it doesn't make sense. I mean, I'm not going to build a steam line out of plastics, right? So I'm going to fix but at the same time, if I have something simple, like a buffer bag to a skid, or a buffer bag to a ferment, or media bag to a fermenter, some things just make more sense today to go disposable than they did, say, 20 years ago.
Beautiful. Well, Ron, we're just out of time here, and we're keeping this on track like a Swiss train. So we can't do much better than quoting Dr. Ian Malcolm from Jurassic Park, and that life finds a way, and so does bioburden. So with that, thank you very much. Great to see you.